Hello, 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 hello. Yes, all right. Everything good and fair and love. So today we are taking a new class which deals about karyokinesis. The cell division came with third cancer karyokinesis. तो आज की हम যেটা পড়তে যাচ্ছি সেটা হলো সেল ডিভিশনের karyokinesis. Cell division में basic mechanism बोलेছিলাম basic mechanism karyokinesis যেটাকে প্রথমে বলে D প্রথমে বলে B phase সাইটোকাইনেসিস C phase দুটোকে মিলে D phase বলে আর এদের বাদে আছে আমাদের যেটাকে বলে ইন্টারফেজ I phase ওকে তো এবারে I phase এর মধ্যে আমরা G1 phase S phase আর এদের মাঝখানে G0 phase এর পরে G2 phase in many phases in many books we see from starting from interphase, but for easy explanation today I'm starting with first karyokinesis. Already I discussed karyokinesis methods and phases like prophase, metaphase, anaphase and telophase. Then too also for a short recap I am repeating this for you things only. So let's start. So let's start karyokinesis. So first of all, we know mitosis. The specific names are given. Specific names are allotted for mitosis. So mitosis is also called mitosis is also called equatorial division or somatic division or mitotic division. Okay, so let's deal with why we say mitosis is also known as equatorial division. So for that, here we know that there is no alteration editing in the chromosomal status of mother cell to daughter cell. So there is a preservation of the chromosomal number between mother and daughter. That is why it is known as equatorial division. And next one, why it is called somatic division? Because it is not happening in a germ cell or a reproductive cell. Then why it is forming? Then it is forming a somatic cell. This type of division is formed at somatic cell of our body, so that's why it is called somatic division. Now it is clear why mitosis is known as equatorial division or somatic division, and every time in board examination this question is being asked repeatedly. So please mark as an important because it seemed to be important for we all, for you all also, for me also. So please mark as underline or mark. This is an important for this. Now let's deal with karyokinesis and cytokinosis. So, actually, I was the main topic on the cytokinosis or interface. Already karyokinesis is correlation तब एक repeatedly video बनाची karyokinesis. So, karyokinesis is a process of nuclear cell division, division of nucleus. Division of nucleus. The process of dividing nucleus in the cell is known as karyokinesis. Okay, so the first phase of a karyokinesis is known as prophase. Prophase is also known as the longest phase. As basically when we see a cell, let's draw. Okay, let's part aside apart. These are the things. Let's draw a cell in detail in a diagram vision. I'm explaining you now. In a vision of diagram, I'm explaining this. So let's imagine this is an animal cell. Now in an animal cell, we know this is a nucleus here with inside. It is a nuclear membrane. Let me tell you why we consider this as an animal cell, because there is no presence of cell wall. And the most important thing, they have no specific shape. They have no specific shape of a cell. Like animal, like plant cell, we see this shape, a rectangular bound shape is there. But here in case there is no specific shape. So in case of animal cell, we can directly say it is animal cell because of these 2 points, these 2 valid reasons. Now let's start. This is a nucleolus. This is a nucleolus, and there is a chromatin network which is consists of DNA material or genetic material from where heredity. The process of heredity starts from the chromatin network of which replicates DNA from mother cell to daughter cell. Okay, clear? Now, another things are also there, like there is a ruby, a ribosome is there now, the endoplasmic reticulum is there, Golgi body is there. Centrosome, most important for cell division in animal cell, is known as centrosome. Centrosome is there. Now centrosome is there. Likewise, mitochondria is also there. Let me draw mitochondria. सेमी ऑटोनोमस ऑर्गेनल के नाम में पहले यहाँ आता है माइटोकॉन्ड्रिया और प्लास्टिक। एक सेमी ऑटोनोमस ऑर्गेनल के नाम आता है क्योंकि इसके पास ओन डीएनए है, ओन डीएनए प्रेजेंस है। ओके, सो हम देखेंगे बाद में इंटरफेस के जीवन फेज में सबसे पहला काम क्या होता है, सेमी ऑटोनोमस ऑर्गेनल के नंबर को मल्टीप्लाई किया जाता है ड्यू टू दिस रीजन ओनली। नाउ आफ्टर दिस इज अ स्पेसिफिक सेल ऑफ अ एनिमल। नाउ इफ वी टॉक अबाउट, नाउ इफ वी Now, if we talk about the next process after this is a prophase, if we start from that. Now, in prophase, let me draw with nucleus first. What we see in nucleus, this is a nuclear membrane. It is a membrane bound organelle. It is a membrane bound organelle we see. So in nucleus there is a membrane, and after that there is a nucleolus, and there is a chromatin network. Many other things present. But in case of this thing in prophase, what we see, first of all, this nuclear membrane gets disintegrate. This nuclear membrane got disintegrate, even though this nucleolus also. After that, this chromatin network is being shortened. Chromatin network converts into chromatin fiber, and after that there will be a non-sister chromatid they formed. They form two non-sister chromatid with the presence of kinetochore and chromatin fiber. Centromere. In prophase only we see this thing, that there is a presence of kinetochore chromatin fiber with the two non-sister chromatid. In the middle there is a chromatid centromere, and with that there is a kinetochore. In prophase this is only. And first thing, before this all, there is the first thing that happens, the viscosity of the cell got increased. Then only we have to get all this thing. So, first thing, or write down in your copy, that the first important thing that happens in prophase. Okay, let me see the name of prophase. So, in prophase it is also known as longest phase. As a dot, mark this. It is also known as longest phase. Why? Because of this all thing that to happening this all thing, it is known as longest phase. So the first point, please write down with me that the viscosity of cell got increased. Next point, nuclear membrane. And nucleolus got disintegrate. After that, chromatin network converted into chromatin fiber. And also here we say that, now let me draw it. First 3 points, write down this thing. First, write down these 3 points. Write down these 3 points first. So, I hope you will write down, copy these 3 points, and prophase known as longest phase in the diagram. Everything in the board after the replicate in your copy, I think. Now, let me see the picture. Let me go back to that picture what we draw in a cell, picture of a cell. that we draw a picture of a cell. This is an animal cell, and here this is two centrosome. There is two centrosome. Now there is a formation of spindle fiber is formed from this thing. The formation of spindle fiber is formed here. Now in prophase only the spindle fiber is formed. Okay. Spindle fiber and astral fiber got formed in prophase. We see now this all thing. That the cell there is many things, but the main focus now goes to this thing itself. So this is a diagram of prophase. If anyone told to identify this picture, then the name the prophase phase of this picture of a subdivision of a kind the mitosis, that is known as prophase, because there is a formation of a, now how I identify? Because there is a formation of spindle fiber, and the most important thing here, the chromosome are not aligned in an equatorial plate, that thing, and there is also the presence of dyad chromosome is there. This is known as dyad chromosome. And because after anaphase, this dyad chromosome gets converted into monad chromosome by longitudinal splitting that we see again. After, but main focus is the most on to this. So let me write down few points of prophase. That the points of prophase are there. Now what is prophase points? The first point is the formation of